Hello friends and welcome back to the channel. It's always nice to have you here. This morning we'll be discussing about methyl red test. Now remember, we are talking about the battery, we are talking about biochemical tests that are used to identify microorganisms in the laboratory, and the essence of all these different tests. Now we've been focused a bit on the IMVIC battery of tests. That is the battery of tests that involves Indo test, Methyl Red test, Vogel's Prostate test, and our citrate test. And these are a battery or a set of tests that we use to identify our members of the family Enterobacteriaceae. And this is important that as I've said earlier, this is important because many members of that family, they are alike in genetic constitution. They are really close. Remember, they're a family of microorganisms. So sometimes it can be difficult to identify them one from the other, but these tests help us to identify them differently. Now, today we are zeroing in on methyl red test, which is one of the tests that we do. Now, methyl red test is a unique test because it's a test, first of all, that helps us to be able to identify if a medium has become more acidic than it used to be before. We know from our elementary science that methyl red is an indicator substance. So it's an indicator substance that helps us to tell pH apart. So it's an indicator substance that will change to a particular color, red in this instance, if the pH is acidic, or pink if the pH is basic or alkaline in nature. Now we use this knowledge to now try to differentiate one organism from another, and it's simple. Special kinds of bacteria are able to undergo what we call mixed acid fermentation. Mixed acid fermentation, and this means that this particular bacteria, when they ferment glucose, when they use glucose as a source of energy and use it up, they are able to generate, as a byproduct of this process, many different types of acids, ranging from acetic acid to formic acid to lactic acid and succinic acid. These four acids are generated as byproducts of this kind of fermentation. Now, not every bacteria can do that. So if you have a bacteria, especially within that family Enterobacteriaceae, the bacteria in that family that is able to undergo mixed acid fermentation, meaning that when they ferment glucose as a source of energy for their respiration, they are able to generate these four different acids that I just mentioned: your lactic acid, your formic acid, your acetic acid, your succinic acid. If they are able to generate this acid, now remember that they are generating in totality four different acids within the same medium. So what happens is that the generation or the production of these four different acids by this organism, when they come in contact with glucose, now significantly lowers the pH of that medium. So if the pH of that medium was neutral, was at 7.0, or maybe even slightly alkaline, the fact that these four different acids have been produced within the same media, it drops the pH of the media. And if the pH of the media is able to drop to something with below about 5.5 to 5.0 to 4.0, then there's a change in the color of any indicator substance. And in this instance, we use methyl red as our indicator substance. So it's simple, and we use it to identify some bacteria different from others within that same family of Enterobacteriaceae. The other kind of bacteria are unable to undergo this mixed acid fermentation, and some other kind of bacteria are only able to, ferment glucose and just produce acetion as an end product of their own process. So just because they're able to produce only acetion, which is not really acidic in nature, they do not cause that change in pH to occur, and thereby they do not, the indicator substance does not change as a result of their own production of acetion. So basically, understand the two different organisms. One set are able to do mixed acid fermentation, produce many acids, drop the pH of the medium, the indicator picks it, and you see it as a color change to red. And if it's the other kind of organism, they produce acetion at the end of their own fermentation of glucose, so they are unable to change pH. That means that the medium remains relatively stable with no change in color. So these are the basics of the of the methyl red test, and it's important. It's an important part, and sometimes and most times it is done in association with the VP test. But we'll talk about the VP test in our next video. So for this test, for this video, we are talking or we are zeroing just on the methyl red test. Now, the process for preparing the methyl red test is that we have what we call methyl red VP broth. So we prepare this broth, and this broth is made up of peptone 7 grams, glucose 5 grams, dipotassium phosphate, and then you have distilled water. Then the pH is adjusted to about 6.9 or 7 before sterilization. Then when all of this is done, we add our indicator substance, our indicator solution, which is methyl red. So we add methyl red, usually 100 ml at of 95% ethanol. And so it's a simple test. When this occurs, then we inoculate with our organism of interest. We inoculate. We also have our control. I've not really been talking about control, which I should be. We have control organisms. So when you run this kind of test, you usually have 2-3 sets, and depending if you are doing it for research, you are running it in triplicates, and then you are also doing your control. And so we inoculate our organism into all of these media, the methyl red VP broth, and then we incubate for at 38 degrees, and we allow for 24 hours. And then when we come back, we look. If we see a notable color change, especially if our indicator has become red, then we realize that the bacteria, it means that the bacteria that we inoculated onto the medium used up the glucose present in that medium and was able to generate or produce 4 different acids, and these acids have dropped the pH of that medium. So it's a really interesting test. We use it to differentiate different results. E. coli is positive, Salmonella is positive, Shigella is positive, but Enterobacter is negative, and Klebsiella is negative. So these are all very important tests that we do. The test, like I said before, is significant for differentiating the different types of organisms. So thank you for listening to this. Remember to like, share our content, and continue to follow us on all our social media platforms. To have a wonderful day.